Wonderful. Hello, guys. Hope that you're good. Today, we are going to talk about eosinophils. These are also very important cells of the immune system involved in several different conditions like allergies and parasitic infections. They have a very important role to play. Um, Unlike neutrophils that we discussed in the last lecture, eosinophils are very few. There would account for 6% of the bone marrow resident nucleated cells, while the neutrophils that we discussed in the last lecture um, were 50 to 70% of all the immune cells. They are the most abundant cells, and neutrophils are the cells that would arrive first at the site of infection because they are so numerous, and they are doing different jobs that we discussed about, and uh, I hope I would upload that lecture as well. um, Eosinophils, when their number exceeds 450 to 500 cells per microliter of the blood, then we call it eosinophilia. And if their number even exceeds further, like 1500 eosinophils per microliter of your blood, then that is called hypereosinophilia. Um, so normally, if you would like to count eosinophils in the blood per microliter, the number of eosinophils should be less than 400. So that's the normal range. Anything about that limit indicates that there is some problem, some kind of parasitic infections, or there may be some allergic conditions in which the eosinophils the number has been raised. And these are, you can see transmission electron micrograph of eosinophil that you see here. You can very easily distinguish eosinophil from a neutrophil. If you look at the neutrophil, it's and the nucleus of the neutrophil, it's multi-lobed. All these granulocytes, they have multi-lobe nuclei, but eosinophil is distinguishable because there are two lobes, that you see here. This is actually the nucleus of the eosinophil. So eosinophils can be distinguished from the neutrophil quite easily by just looking at the structure of its nucleus. If it is a bilobe structure, then definitely it is eosinophil. And if it is a multilobe, more than two lobes, so then that is a neutrophil. And eosinophil, when you see here this transmission electron micrograph, it is filled with these cytoplasmic secretory granules. There are lots of granules inside eosinophil, just like the neutrophils and other granulocytes. And these granules contain four major proteins: eosinophil peroxidase, the major basic protein of MBP, ribonucleases, eosinophil cationic protein, which is called ECP, and eosinophil-derived neurotoxin, or EDN. Now, these proteins have got different roles that we will discuss in the later slide, but uh, these are just the four major basic proteins. But the granules, the contents of the granules are not limited to these different types of proteins that are enzymes that we just learned here, because the granules also store numerous cytokines, uh, enzymes, and growth factors. And other prominent features of eosinophils include primary granules that contain charcoal lead crystal protein, also known as galectin 10, and eosinophil lysophospholipase, and lipid bodies, which are the site of synthesis of cysteine lipotriens, thromboxane, and prostaglandins. So these granules contain a variety of different enzymes and cytokines, And you just should remember that all these are preformed. These are preformed cytokines and enzymes, and these different types of, um, protein molecules inside these granules. These bags are already filled, and the eosinophil has just, it just has to use these different types of enzymes or cytokines when it comes in contact with some kind of stimulus. There are different things that can just trigger eosinophil to, start degranulation or release the contents of these granules or respond differently. There are different ways that eosinophil can respond to different, stimuli or different antigens that we would discuss later. But here you just remember that these are preformed granules, already formed, preformed cytokines and enzymes and different things. And these bags or these granules are filled with all these different types of, uh, factors which can be released when needed. Now, the next slide is, you see here, um, this is a eosinophil, and if you look here, there are receptors for different, things. For instance, there are adhesion receptors like LFA1, CR3, and so many other. And there are receptors for lipid mediators, um, like the platelet activating factor receptor, DP2, DP1, etc. Um, there are cytokine and growth factor receptors for all these different cytokines and growth factors that are mentioned here, like transforming growth factor beta or interleukin 2 receptor, interleukin 3 receptor. So for all these different cytokines and growth factors, the receptors are there on the surface of this particular eosinophil. Similarly, there are receptors for the different bacteria, viruses, fungi, etc., which are known as the pattern recognition receptors. The pattern recognition receptors recognize PAMPs, or pathogen associated molecular patterns. So these patterns are present on different microbes and are recognized by eosinophil with the help of these receptors, which are known as PRRs. So there may be numerous PRRs, like you can see there, the different toll-like receptors, toll-like receptor 1, 2, 3, 4, up to 10, and nod-like receptor, nod 1, nod 2, reg 1, and reg. All these are pattern recognition receptors for recognition of different pathogens on the surface of this eosinophil. Uh, Similarly, there are specific granule contents, and these granules, what would you find inside these granules? All these different cytokines and different cationic proteins, like we have discussed, MBP, EPX, EC, and EDN. Different cytokines and growth factors are also given here. Different chemokines like CCL3, CCL5, etc. And enzymes like acid phosphatases, collagenases, and histaminase, phospholipase, catalase, all these different enzymes and things that we discussed in the last slides. These granules are filled with all these different things. These enzymes, the chemokines, the growth factors, cytokines, and different cationic proteins there for different purposes. The granules of the eosinophils are filled with that. Similarly, there are Fc receptors, and you know the different Fc receptors, C alpha receptor, Fc gamma, or 2 receptors or other receptors here. So these are for different, you can say, antibodies that are recognized here that would bind to those antibodies. And then, the process of recognition of different immune cells, that how they recognize different things with the when it is labeled with some kind of antibody. Then there are lipid, body contents in the form of leukotriene C4, leukotriene D4, leukotriene E4, thromboxane B2, etc., prostaglandins, all these tissue hormones, for instance, these prostaglandins that uh, mm, give us this sense, sensation of pain when you feel there's some inflammatory situation in the body and you feel pain. That is because of the prostaglandins that are, affecting the sensory nerves, and you feel that condition. There's some problem inside your body. Similarly, there are other receptors like MHC class 2. MHC class 2, receptors are important for recognition of different, for presentation of different antigens. For instance, some bacteria or virus or some other pathogen has entered the cell, and the cell has done with it with the help of its enzymes, etc. It has degraded into parts, and the parts of that particular antigen are then displayed on the surface of the cell with the help of MHC class 2 molecules. So it means that this particular cell eosinophil again would be involved in this process of antigen presentation. So there are chemokine receptors. When you look at something like which is starting from CC, it means it's some kind of chemokine. So the CC R1 is a receptor for chemokine 1, CCR3, CCR4. All these are different chemokine receptors, and there are also some other chemokines like receptors for the complements like C5a and C3a. These are the same complements which also trigger uh, you know which can also trigger the mast cells that we discussed in our previous lectures. And these complement 5A and complement 3A also can trigger eosinophils. Um, so when you just look at, this structural diagram, and you look at different things, mentioned here, like different kind of receptor for them, different molecules. You can just you can just do your own assessment that this particular cell would be involved in so many different functions. From each and every receptor, you can just make a story that this particular cell would be doing this kind of job. For instance, this particular receptor, as I told you, has got MHC class 2 receptors, which are specific receptors for the presentation of exogenous antigens like bacteria, virus, etc. So antigen presentation definitely would be done by eosinophils in some conditions when these receptors are present here. This cell can be triggered by all these different things, which has the receptors for which are present on the surface of this. For instance, all these different types of interleukins and growth factors can just, activate this eosinophil to either degranulate or respond in a different way. And all these lipid mediators and chemokines, when or chemokine receptors are there, the chemokines definitely would also, activate this cell, or when the cell, these cells are needed somewhere, because there are very few in the circulation in different tissues, but somewhere when these, cells are needed, then they are recruited with the help of chemo attractants, different things. So these chemoattractant receptors are there on the surface of the cell. So that would be, a kind of attracting eosinophils to, the site of infection or wherever these cells are needed. Uh, these different toll-like receptors that you see here, these are for different pathogens like TLR2 and TLR4, toll-like receptor 2 and toll-like receptor 4, which are, which recognize either proteoglycans in the bacterial wall, or the LPS are lipopolysaccharides. So lipopolysaccharide is a PAM, proteoglycan is a PAM. So all these, different PAMs recognize different toll-like receptors or NOR-like receptors or RIG1. It depends upon the type of PAM, whether it is a nucleic acid and proteoglycan, a lipopolysaccharide, some other content of the pathogenic cell. When these are recognized, then they also can, activate this eosinophil to start with its function. Um, as compared to the neutrophils, you see here is, there's a slight difference here because there are Golgi bodies and endoplasmic reticulum in the eosinophils. In neutrophils, I told you that there are no endoplasmic reticulum. There is no endoplasmic reticulum there, and there are very few Golgi bodies here. And the number in the eosinophils also is, not as much as you would see in the case of other body cells. But still, as compared to the neutrophils, the Golgi bodies and the endoplasmic reticula are comparatively, good in number, and that, the fewer endoplasmic reticulum and Golgi bodies, they there is a different subject that we would discuss in other lectures, that why these cells, have fewer Golgi bodies and endoplasmic reticulum, and how in some cases mitochondria, for instance, in neutrophils, I told you that there are few mitochondria. So what would be the energy dynamics of these particular immune cells, and why these bodies are not, you know, uh, so pronounced inside a particular, immune cell, while there are lots of these bodies in other cells. That is a different subject that we would discuss in some other lecture. Now, the neutrophils, the eosinophils, they are involved in the modulation of the immune system. That is one of, a very important job that the neutrophils would do. The eosinophils would do. For instance, how do a eosinophil modulate, the neutrophils? I told you about these granules, the primary granules in eosinophils, which are filled with different cation proteins and different enzymes and cytokines. So MBP is one of such proteins, cationic proteins, and when MBP is released and there is a neutrophil in the vicinity, the neutrophil is activated, to start making super oxides or secrete interleukin 8 or CR3. So this is a kind of modulating a neutrophil, regulating its responses based on the contents of the eosinophil, something that is released from the granules of, the eosinophil. Similarly, the cytokines which are present in their granules include interleukin 4 and interleukin 13. And when released, both of these cytokines can, act upon a macrophage in the vicinity, and that maintains alternatively activated macrophages in ediphage tissue. I have told you in a separate lecture that macrophages can be activated in a classical way and as an alternate way. So if these cytokines come in contact with the macrophage, they would keep the macrophage in an alternate way of, activation in adipose tissue. And similarly, all these proteins, different proteins that these cationic proteins, if all these proteins come in contact with the mast cells, like the MBP and ECP, EPX, and NGF, um, if there is a mast cell in the surrounding and these proteins act upon the mast cells, then the mast cell would start secreting histamine. And histamine, what is the job of histamine? Histamine increases vascular, uh, it would dilate the vessels, and the vessels dilation is needed when there is some problem in some area of the body, so that the blood supply to that particular area increases. So it also increases the survival of the mast cell. Similarly, if the eosinophil-derived neurotoxin is released from the granules and there is a dendritic cell in the surrounding, this would just, add to the maturation of the dendritic cell, or it would activate the dendritic cell to become mature. Similarly, interleukin 6 in April of the granules, they also increase survival of the bone marrow plasmablast. These contents of the eosinophilic granules, they can also prime the B cells to start, making immunoglobulin M, or these antibodies, the primary antibodies that are made when there's some pathogen that enters your body. So IgM are the first, to be released by the B cells in acute infections. Similarly, they can also, act upon T helper 2 cell types, and, it can, the chemokines are released from the granules can bring about these chemokine chemoattractants can recruit T helper 2 cells to the site of infection. Similarly, it can also act upon other 2 cells, and can do antigen presentation to some extent as well. So you see these are all different immune cells that we are talking about: the macrophages, mast cells, and dendritic cells, neutrophils, helper cells, T cells, B cells. All these cells are modulated, are their responses are controlled, are regulated by contents of the eosinophilic granules. When eosinophils would release these granules, they would bring about modulation of the immune system. That is a very important function of the eosinophils that we are talking about. Um, how these cells originate and how do they migrate into different tissues? Um, Interleukin 3 and GM-CSF and interleukin 5, these cytokines and granulocyte macrophage colony stimulating factors, they have a very important role in the maturation of in the development of eosinophil. Eosinophil, it is a granulocyte, and it would be coming out of the myeloid stem cells in the bone marrow. So the myeloid stem cell is converted into um, mature eosinophil with the help of these growth factors. When interleukin 3 acts upon the myeloid stem cells, and later on interleukin 3 together with GM-CSF, acts upon, the progenitor cells for making these different granulocytes, then the progenitor cells are converted into a eosinophil. So this is, this process of differentiation that these different factors actually are helping with. These different cytokines and GM-CSF, these factors act upon, the myeloid stem cells and the progenitor cells, and, differentiate it along a different line to become a eosinophil. And when the eosinophil is formed in the bone marrow, and this is the bone marrow compartment, then it has to migrate into the blood. And for that migration, something is needed. The eosinophils should be equipped with something, and that is the interleukin 5 receptors. And interleukin 5 receptor is expressed here later on eosinophil, and interleukin 5 receptor, when expressed, the interleukin 5 released in the surrounding would just bind with interleukin 5 receptors. The cytokines receptor is expressed on this cell, and this binds with interleukin 5 in the surrounding, and this binding actually is the key for entry of this eosinophil from the bone marrow into the blood compartment. Now, in the blood compartment, it doesn't stay for longer. So, uh, it would stay here for some time, and then it has to enter different tissues. And in tissues, it has got certain jobs. For instance, it can do a normal physiological job, modulation of the immune system in different organs, or it can aid to the repair of some organs, like support to organ functional integrity, or it can be involved in a disease situation where there are some parasitic or allergic conditions in different organs. And in those conditions, eosinophils are needed. So eosinophils would go into these tissues upon proper invitation, and that would be with the help of different chemokine attractants that are released as a result of some kind of allergic problem or parasitic infection, etc., or some bacterial infection. So the green and the red are two different situations. The green is either a eosinophil involved in immune modulation here in different lymphoid organs of your body, or in other organs, it is involved in support to organ functional integrity. It is involved in the repair process. And in all these different organs, when there's some inflammation or some problem, eosinophils can go upon invitation. Here is, you can say, not a kind of invitation, it's a normal physiological way, for normal physiological reasons, the eosinophil should go there. Then how the eosinophil would enter from the blood compartment, this is the blood compartment, into different tissues. And that happens with, um, with the expression of CCR3 receptors. When this eosinophil has to enter the tissue, after some time, the eosinophils, when they stay for some time in the blood, they would start expression of the CCR3 receptor. And this CCR3 receptors, again, these bind with a number of different chemokines, which includes chemokine L11, chemokine 24 and 26. All these different chemokines can bind with the same receptors, which is the CCR3 receptor. So when this receptor is formed on the surface, these chemokines, in the surrounding, they bind with this receptor. This is a passport for this eosinophil to enter into these different types of tissues. And in these tissues, you see in this this is thymus, spleen, lungs, and gut. These are all lymphoid tissues. Lymphoid tissues means that these organs contains different types of lymphocytes, like B lymphocytes and T lymphocytes. There are sites for the maturation of these B and T cells in these organs. That's why we call them lymphoid organs. So in lymphoid organs, what would a eosinophil do? The eosinophil would do all these different things. These are different lymphocytes that we are talking about. So the lymphocytes would be, uh, modulated, this immune responses would be modulated with the help of eosinophils. Their maturation and activation requires the action of eosinophils, or the contents of eosinophils, the granules that are present there. Then there is you can say, in some organs, like in uterus, normally you would see eosinophils in the uterus, and similarly eosinophils also contribute a great deal towards the development of the mammary glands and its maintenance, and the adipose tissue you would find eosinophils. So in these organs, eosinophils are involved in maintaining the functional integrity. So it's not only immune modulation, also the functional integrity, or which are the normal jobs of the eosinophils that they do in these different organs that are mentioned here. And when there's some kind of inflammation, I have already told you, in all these organs like the skin, lungs, nasal mucosa, heart, blood vessels, etc., eosinophils can enter upon invitation when these chemokines are released as a result of some problem. The eosinophils would enter into any of these tissues and perform their jobs. In some conditions, you can just diagnose this, whether there is infiltration of the eosinophils in heart tissues or some other tissues, and you can also go for diagnosis of different conditions. So these are different proteins, different uh, cationic proteins that we discussed about, um, and the galactin-10 CLC protein, and their functions, that what are they doing actually, and you can just go through the role of these different proteins, some of which are um, already have been discussed. Now, eosinophils, they would do phagocytosis, cell killing, and antigen presentation at the same time. All these for different functions that the eosinophil is doing. Eosinophils are able to phagocytose invading pathogens to some extent. The neutrophils are very actively doing that particular job, but eosinophils also can do that job. You can say that they are not very actively doing that job. They are, able to do phagocytosis of antigens and degradation of antigens, but to a certain extent, and kill them intracellularly by delivering these cationic proteins like the MBP and ECP, which are delivered to the intracellular phagosome when something enters, and a phagosome is formed inside. So these MBP and ECP cationic proteins are delivered into that phagosome, which contains that bacterium or virus, and it is degraded with the help of these things. This in turn paves the way to subsequent antigen presentation. And I told you about MHC class II molecules, which are expressed on the surface of eosinophils. So the eosinophils are also capable to present that particular parts of antigen on surface in order to alert other immune cells to come here and recognize that antigen, which is coupled with MHC class II on its surface. And that's the normal immune responses that you already know. In addition, eosinophils are also endowed with extracellular killing mechanisms, which includes releasing cytotoxins through degranulation, performing a respiratory burst through EPO, as well as extracellular DNA trapping. Uh, you know, this is a very unique property of the eosinophils, that they can also it's not necessary to phagocytose an antigen and then a bacterium or a virus, and then chop it into pieces and present it on the surface of it. This particular cell, just like the neutrophils that I told you, can also uh, kill the antigens extracellularly without phagocytosing it. For instance, it can release these different cytotoxins which are present there inside its granules, and with the help of these cytotoxins can just degrade that particular pathogen there, which later would be taken up by different types of macrophages in your system. or through respiratory burst or through extracellular DNA trapping that I told you that um, there is this property in the neutrophils as well, recently discovered in eosinophils as well, that its cellular chromatin can just protrude out of the cell, and it's associated with different enzymes, and this when these DNA or you can say these chromatin fragments protrude out of the eosinophils, it makes a trap, and the pathogens traveling around are just trapped in this chromatin, which is associated with different enzymes in your circulation or in different tissues, and those enzymes just degrade, uh, the pathogens. So this is a very unique extracellular killing mechanism in eosinophils. Degranulation in eosinophil is tightly regulated. In most cases, small quanta of selected cytotoxins from the specific granules are released in the extracellular space. It is known as piecemeal, degranulation, when small amounts are released in the space instead of full blown degranulation. So that is a highly regulated process, not like something random. Uh, that was about eosinophils, that how where the eosinophils originate and how do they migrate in different tissues, and for what purposes, what are the functions that these cells are doing, and how the eosinophils are doing immune modulation. You can just uh, um, go to the details by looking at the slide, and here are different receptors that also are telling you the stories, uh, uh, different stories, like about the eosinophils, that how many different types of functions the eosinophils would be involved in by just looking at the slide, and different kind of receptors that are present on its surface. So they are involved in so many different types of functions. So it was about the eosinophils. Hope to see you later in some other lecture about other immune cells. Thank you very much for listening to this. Hope to see you again.